8.5.3 Endomembrane system. While each of the membranous organelles is distinct in terms of its structure and function, many of these are considered together as an endomembrane system because their functions are coordinated. The endomembrane system include endoplasmic reticulum, which is ER, Golgi complex, lysosomes and vacuoles. Since the functions of the mitochondria, chloroplast and peroxisomes are not coordinated with the above components, these are not considered as part of the endomembrane system. 8.5.3.1 The Endoplasmic Reticulum ER. Electron microscopy studies of eukaryotic cells reveal the presence of a network or a reticulum of tiny tubular structures scattered in the cytoplasm that is called the endoplasmic reticulum ER, figure 8.5. Hence, ER divides the intracellular space into two distinct compartments, that is, a luminal inside ER and extraluminal cytoplasm compartments. The ER often shows ribosomes attached to their outer surface. The endoplasmic reticulum bearing ribosomes on their surface is called rough endoplasmic reticulum RER. In the absence of ribosomes, they appear smooth and are called smooth endoplasmic reticulum SER. RER is frequently observed in the cells actively involved in protein synthesis and secretion. They are extensive and continuous with the outer membrane of the nucleus. The smooth endoplasmic reticulum is the major site for synthesis of lipid. In animal cells, lipid-like steroidal hormones are synthesized in SER. 8.5.3.2 Golgi apparatus. Camillo Golgi 1898 first observed densely stained reticular structure near the nucleus. These were later named Golgi bodies after him. They consist of many flat disc-shaped sacs or cisternae of 0.5 micrometer to 1.0 micrometer diameter, figure 8.6. These are stacked parallel to each other. Varied number of cisternae are present in a Golgi complex. The Golgi cisternae are concentrically arranged near the nucleus with distinct convex cis or the forming phase and concave trans or the maturing phase. The cis and the trans phases of the organelle are entirely different but interconnected. The Golgi apparatus principally performs the function of packaging materials to be delivered either to the intracellular targets or secreted outside the cell. Materials to be packaged in the form of vesicles from ER fuse with the cis phase of the Golgi apparatus and move towards the maturing phase, which is the trans phase. This explains why the Golgi apparatus remains in close association with the endoplasmic reticulum. A number of proteins synthesized by ribosomes on the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus before they are released from its trans phase. Golgi apparatus is the important site of formation of glycoproteins and glycolipids. 8.5.3.3 Lysosomes. These are membrane-bound vesicular structures formed by the process of packaging in the Golgi apparatus. The isolated lysosomal vesicles have been found to be very rich in almost all types of hydrolytic enzymes, hydrolases, lipases, proteases, carbohydrases, optimally active at the acidic pH. These enzymes are capable of digesting carbohydrates, proteins, lipids, and nucleic acids. 8.5.3.4 Vacuoles. The vacuole is the membrane-bound space found in the cytoplasm. It contains water, sap, excretory product, and other materials not useful for the cell. The vacuole is bound by a single membrane called tonoplast. In plant cells, the vacuoles can occupy up to 90% of the volume of the cell. In plants, the tonoplast facilitates the transport of a number of ions and other materials against concentration gradients into the vacuole, hence their concentration is significantly higher in the vacuole than in the cytoplasm. In amoeba, the contractile vacuole is important for osmoregulation and excretion. In many cells, as in protists, food vacuoles are formed by engulfing the food particles. 8.5.4 Mitochondria. Mitochondria, singular mitochondrion, unless specifically stained, are not easily visible under the microscope. The number of mitochondria per cell is variable depending on the physiological activity of the cells. In terms of shape and size also, considerable degree of variability is observed. Typically, it is sausage-shaped or cylindrical, having a diameter of 0.2 to 1.0 micrometer, average 0.5 micrometer, and length 1.0 to 4.1 micrometer. Each mitochondrion is a double membrane-bound structure with the outer membrane and the inner membrane dividing its lumen distinctly into two aqueous compartments, that is, the outer compartment and the inner compartment. The inner compartment is filled with a dense homogeneous substance called the matrix. The outer membrane forms the continuous limiting boundary of the organelle. The inner membrane forms a number of enfoldings called the cristae, singular crista, towards the matrix figure 8.7. The cristae increase the surface area. The two membranes have their own specific enzymes associated with the mitochondrial function. Mitochondria are the sites of aerobic respiration. They produce cellular energy in the form of ATP, hence they are called powerhouse of the cell. The matrix also possesses singular circular DNA molecule, a few RNA molecules, ribosomes 70S, and the components required for the synthesis of proteins. The mitochondria divide by fission. 8.5.5 Plastids. Plastids are found in all plant cells and in euglenoids. These are easily observed under the microscope as they are large. They bear some specific pigments, thus imparting specific colors to the plants. Based on the type of pigments, plastids can be classified into chloroplasts, chromoplasts, and leucoplasts. The chloroplasts contain chlorophyll and carotenoid pigments which are responsible for trapping light energy essential for photosynthesis. In the chromoplast, fat-soluble carotenoid pigment like carotene, xanthophylls and others are present. This gives the part of the plant a yellow, orange or red color. The leucoplasts are the colorless plastids of varied shapes and sizes with stored nutrients, which is amyloplast store carbohydrates, that is starch, example potato. Elioplast store oils and fats, whereas the aleuroplast store proteins. Majority of the chloroplast of the green plants are found in the mesophyll cells of the leaves. These are lens-shaped, oval, spherical, discoid or even ribbon-like organelles having variable length up to 5 to 10 micrometer and width up to 2 to 4 micrometer. Their number varies from 1 per cell of the Chlamydomonas, a green alga, to 20 to 40 per cell in the mesophyll. Like mitochondria, the chloroplasts are also double membrane bound. Of the two, the inner chloroplast membrane is relatively less permeable. The space limited by the inner membrane of the chloroplast is called the stroma. A number of organized flattened membranous sacs called the thylakoids are present in the stroma, figure 8.8. Thylakoids are arranged in stacks like the piles of coins called grana, singular granum, or the integral thylakoids. In addition, there are flat membranous tubules called the stroma lamellae connecting the thylakoids of the different grana. The membrane of the thylakoids enclose a space called a lumen. The stroma of the chloroplast contain enzymes required for the synthesis of carbohydrates and proteins. It also contains small double-stranded circular DNA molecules and ribosomes. Chlorophyll pigments are present in the thylakoids. The ribosomes of the chloroplast are smaller 70S than the cytoplasmic ribosomes 80S. 8.5.6 Ribosomes. Ribosomes are the granular structures first observed under the electron microscope as dense particles by George Pallet 1953. They are composed of ribonucleic acid RNA and proteins and are not surrounded by any membrane. The eukaryotic ribosomes are 80S while the prokaryotic ribosomes are 70S. Each ribosome has two subunits, larger and smaller subunits, figure 8.9. The two subunits of 80S ribosomes are 60S and 40S, while that of 70S ribosomes are 50S and 30S. Here S stands for Swedberg's unit, stand for the sedimentation coefficient. It is indirectly measure of density and size. Both 70S and 80S ribosomes are composed of two subunits. 8.5.7 Cytoskeleton. An elaborate network of filamentous proteinaceous structures consisting of microtubules, microfilaments, and intermediate filaments present in the cytoplasm is collectively referred to as the cytoskeleton. The cytoskeleton in a cell are involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. 8.5.8 Cilia and Flagella. Cilia singular cilium and flagella singular flagellum are hair-like outgrowth of the cell membrane. Cilia are small structures which work like oars, causing the movement of either the cell or the surrounding fluid. Flagella are comparatively longer and responsible for cell movement. The prokaryotic bacteria also possess flagella, but these are structurally different from that of the eukaryotic flagella. The electron microscopic study of a cilium or the flagellum show that they are covered with plasma membrane. Their core, called the exoneme, possess a number of microtubules running parallel to the long axis. The exoneme usually has 9 doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules. Such an arrangement of exonomal microtubules is referred to as the 9 plus 2 array, figure 8.10. The central tubules are connected by bridges and is also enclosed by a central sheath which is connected to one of the tubules of the each peripheral doublets of a radial spoke. Thus, there are 9 radial spokes. The peripheral doublets are also interconnected by linkers. Both the cilium and flagellum emerge from centriole-like structure called the basal bodies. 8.5.9 Centrosome and Centrioles. Centrosome is an organelle usually containing two cylindrical structures called centrioles. They are surrounded by amorphous pericentriolar materials. Both the centriole in a centrosome lie perpendicular to each other, in which each has an organization like the cartwheel. They are made up of 9 evenly spaced peripheral fibrils of tubulin protein. Each of the peripheral fibril is a triplet. The adjacent triplets are also linked. The central part of the proximal region of the centriole is also proteinaceous and called the hub, which is connected with tubules of the peripheral triplets by radial spokes made of protein. The centrioles form the basal body of cilia or flagella and spindle fibers that give rise to spindle apparatus during cell division in animal cells. 8.5.10 Nucleus. Nucleus as a cell organelle was first described by Robert Brown as early as 1831. Later, the material of the nucleus stained by the basic dyes was given the name chromatin by Fleming. The interphase nucleus, that is nucleus of a cell when it is not dividing, has a highly extended and elaborate nucleoprotein fibers called chromatin, nuclear matrix and one or more spherical bodies called nuclei, singular nucleolus. Figure 8.11. Electron microscopy has revealed that the nuclear envelope, which consists of two parallel membranes with a space between 10 to 50 nanometer called the perinuclear space, forms a barrier between the materials present inside the nucleus and that of the cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and also bears ribosomes on it. At a number of places, the nuclear envelope is interrupted by minute spores which are formed by the fusion of its two membranes. These nuclear pores are the passage through which movement of RNA and protein molecules takes place in both directions between the nucleus and the cytoplasm. Normally, there is only one nucleus per cell. Variations in the number of nuclei are also frequently observed. Can you recollect names of organisms that have more than one nucleus per cell? Some mature cells even lack nucleus, example erythrocytes of many mammals and sieve tube cells of vascular plants. Would you consider these cells as living? The nuclear matrix or the nucleoplasm contains nucleolus and chromatin. The nucleoli are spherical structures present in the nucleoplasm. The content of nucleolus is continuous with the rest of the nucleoplasm as it is not a membrane bound structure. It is a site for active ribosomal RNA synthesis. Larger and more numerous nuclei are present in cells actively carrying out protein synthesis. You may recall that the interphase nucleus has a loose and indistinct network of nucleoprotein fibers called chromatin. But during different stages of cell division, cells show structured chromosomes in place of the nucleus. Chromatin contains DNA and some basic proteins called histones, some non histone proteins, and also RNA. A single human cell has approximately 2-meter long thread of DNA distributed among its 46 23 pairs chromosomes. You will study the details of DNA packaging in the form of a chromosome in class 12th. Every chromosome, visible only in dividing cells, essentially has a primary constriction or the centromere on the sides of which disc-shaped structures called kinetochores are present, figure 8.12. Centromere holds two chromatids of a chromosome. Based on the position of the centromere, the chromosomes can be classified into four types. Figure 8.13. The metacentric chromosome has middle centromere forming two equal arms of the chromosome. The submetacentric chromosome has centromere slightly away from the middle of the chromosome, resulting into one shorter arm and one longer arm. In case of acrocentric chromosome, the centromere is situated close to its end, forming one extremely short and one very long arm, whereas the telocentric chromosome has a terminal centromere. Sometimes a few chromosomes have non-staining secondary constrictions at a constant location. This gives the appearance of a small fragment called the satellite. 8.5.11 Microbodies. Many membrane bound minute vesicles called microbodies that contain various enzymes are present in both plant and animal cells.